There are several differences between prokaryotic and eukaryotic transcription. For one, transcription is much more of a needle in a haystack problem than prokaryotic transcription. In prokaryotes, there is one gene every 1,000 base pairs. In the human genome, we have about 20,000 genes in a genome that's 3 billion base pairs long, and that means human genes are found once in 150,000 base pairs. And so genes are much more spread apart in eukaryotic genomes than they are in prokaryotic genomes, primarily because prokaryotic genomes tend to be very small and compact. Perhaps because of this, eukaryotic transcription is more complex. The initiation is more complex. And whereas in prokaryotes there's just one RNA polymerase, in eukaryotes there are 3. RNA polymerase 2 or pol 2 transcribes messenger RNA. Pol 1 is specialized in transcribing ribosomal RNA or rRNA, and pol 3 specializes in transcribing transfer RNA, small nuclear RNAs, as well as the 5S ribosomal RNA. A second difference in eukaryotic transcription from prokaryotes is that eukaryotic cells have compartments. So you have nuclei which contain the genome, and that's where transcription happens. And then you also have cytoplasm, which is where the ribosomes are and translation happens. And that means transcripts need to be exported, and that necessitates processing of the transcripts that you have to do in eukaryotic transcription, but you didn't have to do in prokaryotic transcription. And the third major difference between eukaryotic and prokaryotic transcription is that in eukaryotic transcription you have to think about chromatin. Although chromatin is more involved in the regulation of transcription than transcription per se. However, that's one of the differences between eukaryotic and prokaryotic transcription. The stages of transcription are the same in eukaryotes. You have initiation, elongation, and then termination. The initiation and the polymerase holoenzyme complex is a lot more complicated. It starts off with what's known as the TATA box, which contains a sequence pattern, a sequence motif, TATA. That's a TATA box, which is about 30 base pairs before the transcription start site. And this is also called upstream, since if the transcript is a river. It flows 5' to 3' in this direction. So this is upstream. Everything occurring before the transcript is upstream, and after the transcript is downstream. So 5' of the transcript is upstream and 3' of the transcript is downstream. And this TATA box is about 30 base pairs upstream of the transcription start site. And to this TATA box binds a protein known as TATA binding protein, or TBP, followed by a protein called TF2D. And these initiation factors are called general transcription factors, or GTFs. And following the recruitment of TF2D, more GTFs are recruited: TF2I, TF2H, and TF2F. And at this point, the polymerase starts to elongate, and one of the GTFs phosphorylates the carboxy terminal domain, or the C terminal domain of the RNA polymerase, which has important roles in the processing of transcripts. And there are several processing steps that are involved. The first one is the addition of what's known as the 5' cap by the capping enzymes. And the 5' cap is a 7-methylguanosine. And so a guanosine, so a guanine with the ribose sugar, which is guanosine, which has been methylated on the 7th carbon. This 7-methylguanosine is connected via 3 phosphates, 5' to 5'. So the 5th carbon of the 7-methylguanosine, of the ribose of the 7-methylguanosine, is bonded to the 5th carbon of the first nucleotide of the mRNA, or the transcript molecule. And this 5' cap protects the transcript from degradation. The cell has several mechanisms for a defense against viruses, and one of those mechanisms is to degrade RNAs, since retroviruses have their genetic material in the form of RNA, and the legitimate bona fide RNA being made by the cell, you add a 5' cap, which is a signal to RNA degradation machinery to not degrade this host RNA. The next processing step is that of splicing, and eukaryotic mRNAs can be composed of different sections where the sections called exons are translated and code for polypeptides, but these exons are interspersed with stretches called introns that are cut out or spliced out and are never translated into polypeptide. And we will look at splicing in greater detail in a bit, but the importance of splicing is that it allows you to generate a large number of different types of proteins by combining different exons in different combinations, and that's one of the steps, processing steps, that must occur during eukaryotic transcription. And the third processing step occurs after termination when a poly A tail, so a string of about 150 adenosines is added to the 3' end of the transcript, and the function of the poly A tail is twofold. One is, like the 5' cap, to protect the transcript from degradation, and secondly, to help in nuclear export, to export the transcript out of the nucleus. The 3' UTRs of eukaryotic transcripts, so let's say this is the 5' UTR, and this is the 3' UTR. They contain a sequence signature, a particular sequence called the poly A signal, and that is either AAU, AAA, and this is in the transcript, and on the coding strand it would be AATT, AAA, or it could be AUU, AAA. And when the polymerase encounters the poly A signal, it terminates transcription about 20 base pairs downstream of the poly A signal, and then it adds the poly A tail, which is about 150 to 200 base pairs of adenine bases. So this is the poly A tail, and this is the poly A or pause signal. Next, splicing is the process by which introns are removed or cut out or spliced out from eukaryotic RNAs. In this example of the rat alpha tropomyosin gene, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 introns, and 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 exons. And these exons, once the introns have been spliced or cut out, can be combined in many alternative conformations. For instance, this particular transcript called brain TMBR2 has exons 1B, 3, 4, 5, 6B, 7, 8, and 9B, whereas in this fibroblast TM2 transcript has 1A, 2B, 3, 4, 5, 6B, 7, and 8. And this is called alternative splicing, which refers to the fact that you can splice out the introns in different ways. Moreover, not only is it possible to have different forms or isoforms of the transcript, different isoforms are found in different cell types or tissues. So, for example, smooth muscle has these two isoforms, whereas brain has 3 of these isoforms, and so on and so forth. And therein lies the significance of splicing and alternative splicing, which is that it gives us a much greater diversity of isoforms of proteins that can be produced from the same number of genes. And the second important aspect is that it can create alternative splicing can create specialized behaviors in different cell types by creating alternative proteins with distinct functions. So how does splicing work? The border between the intron and the exon are called the 5' splice site and the 3' splice site. And the sequence which is near the splice site is highly conserved. So you have a GU 100% of the time and AG 95% of the time. The 3' splice site has an AG 100% of the time, a C 80% of the time, and there is, at a distance of about 15 to 45 bases, with 100% probability an A, and this A is called the branch point. And this pattern of sequence at the splice sites is used by the splicing machinery to recognize introns and to remove them. This machinery is called the spliceosome. And it consists of small nuclear ribonucleoprotein complexes, and these small nuclear ribonucleoprotein complexes, or SNRPs, are composed of small nuclear RNAs plus proteins. And we will consider 6 of them, U1 through U6, that are involved in splicing. So the U1 snRNP binds the 5' splice site, whereas the U2 snRNP binds the branch point. At this point, U4, U5, and U6 snRNPs are recruited and kind of bind the intron and cause it to fold, and this causes the release of U1, U4, and U6 snRNPs. At this point, the branch point adenine is going to undergo a transesterification reaction, which will link the 2' carbon of the adenine at the branch point to the 5' carbon of the G at the 5' splice site through an ester bond. And this transesterification, once it's complete, will do 2 things. First, the 3' carbon of the 3' end of exon 1 will be free, and because of the transesterification reaction between the 2' carbon of the A at the branch point and the 5' carbon of the G at the 5' splice site, you will form this lasso structure, and this is called a lariat. And in the next step, since the 3' carbon has a hydroxyl, it's no longer linked in a chain of nucleotides, it can undergo a transesterification reaction with the G at the 3' splice site, so that you can form a phosphodiester bond and thereby connect the 2 exons together and release the lariat of the intron.